Good afternoon, students. So, good afternoon, all of you. So, welcome to Unacademy EAMCET YouTube channel, students. So, welcome to Unacademy EAMCET YouTube channel. So, good afternoon once again. So, myself, Dr. Rajendra Prasad. So, biology educator on Unacademy platform, and I have been teaching biology to NEET and medical aspirants from last 13 years. So, in today's class, we are going to discuss about the EAMCET 2020 question paper. So from last few classes I have been discussing EAMCET 2020 question papers. The same thing we are going to do in today's session also. So we will discuss 20 questions. We will discuss the 20 questions of the EAMCET 2020 question paper. So that was conducted on the 24th of September. So before starting the class, so I would like to introduce about our Unacademy Plus subscription. So I would like to continue about the Unacademy Plus subscription, students. So Unacademy is providing free live classes. So Unacademy is providing free live classes every day on its YouTube channel, that is Unacademy EAMCET YouTube channel, students. Apart from that, Unacademy is also providing free special live classes on its app. So apart from the free YouTube classes, you can also join the Unacademy free special classes. So that is conducted on our website. So there is a link is provided in the description box. You can check the description box of this particular class. In that there is a links are provided so that you can directly open the Unacademy app link and you can join the free special live classes of all the subjects, maths, physics, chemistry and as well as biology. Apart from that, so there is Unacademy Plus. So that is another platform, so where a well designed and a scheduled course is going on for completion of the syllabus for students who are preparing for next year medical entrance examination. It is a well designed course. So it is a certainly benefit for the students. So that is a Unacademy Plus subscription. So let us see what is the benefits of Unacademy Plus subscription. So Unacademy Plus subscription, what happens, a designed course is going on to complete the syllabus of 11th class and 12th class syllabus, so that student can get ready by the next end of the in 2020. By end of the March, you will be getting ready for the examination, for your medical entrance examination. So I suggest you to take the Plus subscription, students. So a personal care will be taken by the respective teachers. You can doubt clearing sessions also will be available. So what is the importance of the Plus subscription is a premium content. So premium content will be available at affordable price. So structured live courses and dedicated loud clearing session is also will be taking place. After every 3 classes, 4th class will be a doubt clearing session, so that you can interact with your educator and you can ask and clear the doubts. So what type of doubts you will be having, so you can clear from it. So that is the benefit of Plus subscription. So quizzes also will be conducted at regular intervals. So minor quizzes and major quizzes also will be available. So and lecturers on this platform of Unacademy EAMCET are divided into batches. You can enroll those batches which you are interested, students. So by taking the Plus subscription, so test series and full and mock tests are also being conducted. So if you are interested, you can use my code RAJENDRALIVE so that you can get all these courses for 1 month plan, that is of a trial plan, that is 2,250 rupees, students. So on the app it will be showing 2,500 per 1 month. If you use my code RAJENDRALIVE, so if you use my code RAJENDRALIVE, you can get the course for 2,250 rupees. So that is the price of the Unacademy Plus subscription for 1 month, students. There is one batch is started, that is called as a Telugu batch. So in this batch, a set of lectures are there. You can see that biology, physics, mathematics, chemistry and physics lecture. They will be taking classes on Plus platform only in our native language, that is in Telugu. So they will be teaching everything in Telugu. So detailed explanation will be given in Telugu language. That is benefit of this particular batch course. So their target is also, the lecture's target is also to complete syllabus by of intermediate first year and second year and make the students get ready for next year EAMCET 2021. So complete syllabus will be covered. So that is benefit of this particular plan. So tips and tricks also will be given by educators. So this batch is recently started in the October month. So maybe 1 or 2 lessons there may be in progress. So you can join this batch. So another batch, there is another batch which I am taking the biology classes, and sir will be taking the physics classes, and this sir will be taking the chemistry classes. So the main purpose of this batch is called as a comprehensive batch. So main purpose is also to complete syllabus by end of the February month. Tips and tricks will be given by top educators, no doubt about it. So topic wise practice papers also will be given, and full and mock tests also will be given to the students. Certainly it is benefited. So 1 month plan you can purchase if you are interested. So you can use my code RAJENDRALIVE so that you can get a 1 month plan for 1250 rupees. Means for that we will be getting discount. So 2250 rupees you can get. So I suggest you to take long term plan, students. So there is a long term plan for 6 months and 12 months. So 6 months plan, if you take 6 months plan, it is costing around 10,125 rupees, and there is another best plan, that is 12 months plan. So it is costing 12,375 rupees. So these are the two best plans which I suggest you to take, so that you can get the benefit out of this. So whole syllabus will be covered on this plus subscription platform, students. So I suggest you to take the Unacademy long term plans of 6 months plan as well as the 12 months plan, then only certainly you will be getting benefited out of this. So no doubt here also, so everywhere, so plus, if you take plus subscription in any batch course, the premium content will be given, and structured live course and dedicated live outline sessions will be present, and test series also will be conducted, students. So all this you can get for a single subscription of unlimited access. So that is our 6 months plan 10,125 rupees and 12 months plan of 12,375 rupees. So that is about our Unacademy plus subscription, students. So I suggest all students to join plus subscription so that you can get benefit out of this, and very less amount of 10,000 rupees for 6 months is a very less amount. So no doubt about it, you can compare with your corporate college fees also. So let us discuss the multiple choice questions in this class, which we are going to discuss now. So these are the questions which that came on the 24th September of EAMCET examination. So let us see the questions one by one. So let us see this question. So transpiration plays a significant role in the concept of, so transcription is playing a significant role. So what is the meaning of this transpiration? So what is the meaning of this transpiration? If you see, it is the loss of water. So what is the transpiration? Loss of water. So loss of water, okay, from the plant. So loss of water from plant in the presence of sunlight. So loss of water is taking place from the plant in the presence of sunlight. So in the presence of sunlight. So that process is called as, a transpiration. So loss of water. So this loss of water is taking place, okay. So this plays a significant role. So what happens when there is loss of water? We already know that first plant absorbs water from the soil. Inside the soil, water is present. Water will be absorbed by the plant. So that water will be absorbed by the plant through roots. From the roots, it will be reaching the stem. From stem, it will be reaching to the leaves. In the leaves, the water will be released out through stomata through a process that is called transpiration. So because of this, what happens is there is a significant role is playing. That is, then there is an interaction between the soil, plant, and as well as atmosphere. So physical, like light, sunlight is more present, means more transpiration. So there is an association. So there is a association between soil, plant, and as well as atmosphere, especially the sunlight. So that is the reason we have to take option 4 as our answer. So the interaction is taking place between the soil, because inside the soil we have the water. So that water is taken by the plant, and from the plant, so due to excess sunlight, what happens is evaporation of water is taking place. So that process is called as a loss of water. It is called evaporation of water. So this evaporation of water from the leaves of the plant, so that process is called as a transpiration. So finally, so significant role. So transpiration plays a significant role in concept is soil, plant, atmospheric continuum. So that will be the answer. Fourth option will be our answer, students. Okay. So that is about that question. So let us move to the next question. So let us see the next question, students. So dumbbell shapes. So dumbbell shaped guard cells. So just now we are discussing the guard cells. So guard cells play important role in the transpiration. So just now we are discussing about transpiration. So water is moving out of the plant through the leaf. We were discussing the same thing. So inside the leaf, if you say take the internal structure of the leaf, inside the leaf we have epidermis. On the epidermis we have these guard cells. So these guard cells, if you take the guard cells, so no doubt these guard cells, so they are present in the leaf of dicot leaf as well as in the guard cells. Where they are present means they are present in the leaf. So they are present in the dicot leaf as well as in the monocot leaf, no doubt about it. So if you see the shape of the guard cells, in case of the dicot leaf, so guard cells are bean shape. So in case of the in case of the dicot leaf, in dicots what happens is the guard cells are bean shape, or they are also called as a kidney shape. They are bean shape, or they are also called as a kidney shape. So whereas in case of monocots, especially the monocots like grasses, so monocots like grasses, in case of the monocots like grasses, they appear as a dumbbell shape. So they appear as a dumbbell shape. So that is the shape of the guard cells. So the guard cells plays a main role in the evaporation process. They open, and so that when they open, the loss of water will be taking place. So when there is a turgidity, guard cells open. When guard cells become turgid, what happens is guard cells open and loss of water is taking place, and that process is called as a transpiration. Anyhow, so the guard cells are present in both dicot leaf and as well as monocot leaf, especially in the epidermis region. So they are present in the epidermis region. So the shape of the guard cells, in case of the dicot leaf, the shape of the guard cell is a bean shape, whereas, or it is called as a kidney shape, whereas in case of the monocot leaf, the shape of the guard cells is it is a dumbbell shape. So based on that, we have to take the answer. So anyhow, so our answer will be the, so no doubt it is in the grasses, it is a dumbbell shape. So our answer will be the option 4, that is the grasses will be our answer. So that is about this question. Let's move to the next question, students. So let's see the next question. So the main difference between gram positive bacteria and gram negative bacteria. So this prokaryote, so bacteria prokaryote, bacteria is no doubt it is a prokaryote. This bacteria is classified. So bacteria is further classified into two types. So one type is called as a gram positive bacteria. So another one is called as a gram negative bacteria. So bacteria is classified into gram positive and gram negative based on the cell wall composition. So that is the important point which you have to remember. So based upon the cell wall composition, the bacteria is classified into gram positive and gram negative. So based upon cell wall composition. So if you see what is the difference, major difference. So gram positive bacteria is having thick peptidoglycan. So there is a thick peptidoglycan is present. So this is the major difference. So in case of gram negative bacteria, it is having a thin peptidoglycan layer. So it is having a thin peptidoglycan. So thin peptidoglycan layer. So that is the major difference actually. So that is the major difference. So the bacteria is classified by Christian Gram. So Christian Gram is the name of the scientist. He has done a technique. He has introduced a technique that is called gram staining. So by using that gram staining technique, he has differentiated bacteria based upon cell wall composition into gram positive bacteria as well as gram negative bacteria. Then what is the difference between gram positive and gram negative bacteria? Gram positive bacteria is having thick peptidoglycan layer, whereas gram negative bacteria is having a thin peptidoglycan layer. So that is a major difference. So based on that, we have to identify our answer. So the main difference between gram positive and gram negative bacteria, no doubt it is a cell wall. So cell wall is the major difference. So that is our answer. So cell wall will be our answer, students. So let us move to the next question. So that is about this question. Let us move to the next question. So next question, let us see the next question. S phase. Question is asking about the S phase. So S phase of cell cycle. So if you take this cell cycle, so in that cell cycle we have interphase. So we have interphase and as well as we have M phase. So interphase and as well as M phase. So this interphase. So interphase is divided into G1 phase, S phase and G2 phase. So M phase. So this M phase is divided to prophase, metaphase, anaphase and as well as telophase. So like that. So question is asking about the S phase. So you can see this is the S phase. S phase is a part of the interphase. So what is the main role of the S phase? In S phase what happens? So in S phase what happens is the DNA replication takes place in the S phase. Okay. So based on that, let us see the options. First option, you see DNA doubles per cell. So no doubt about it, DNA is doubling. Then number of chromosomes will not decrease. Number of chromosomes remain same. So Number of chromosomes increase, so this also cannot be answered. Number of chromosomes remain same actually. So synthesis of protein also does not takes place. Synthesis of proteins takes place in G2 phase. Synthesis of proteins takes place in the G2 phase. So what is this S phase? Means it is a part of interphase. And what is the main event that is taking place in S phase? In S phase, what happens is DNA replication is taking place. In S phase, DNA replication is taking place in case of the S phase. So DNA replication is taking place in the S phase, no doubt about it. So DNA replication, it is taking place in the S phase, is DNA replication. So DNA replication means doubling of the DNA is called as a DNA replication or DNA duplication. So based on that we have to identify. So option 1 will be our answer based on that. Okay. So that is about this particular question, that is S phase. So welcome to Yashwant. So good afternoon Yashwant. So welcome to Unacademy EAMCET YouTube channel. So we are discussing the EAMCET questions. So no doubt your answer is correct, no doubt A is the answer. Okay, let us move to the next question. Choose the correct pair of cell wall. So choose the correct pair of cell wall. So choose the correct pair of cell wall degrading or cell wall digesting enzymes. So cell wall digesting enzymes. So let us see the lipase. Let us see with the option 4. Let us start with the option 4. So lipase. So what is the main function of lipase? So lipase main function, if you see the lipase main function is, so it is nothing but it is breaking off or breaking or it is digesting. We should say not breaking actually, we should say digesting actually, or hydrolyzing. Okay, digesting of the lipids. So lipids are broken down by lipids. So lipids are broken down by enzyme lipase. So cellulose, no doubt it is not an enzyme, it is a polysaccharide. Cellulose is a polysaccharide. So cellulose is a polysaccharide. So in fungus, okay, so fungus is having chitin but not the cellulose. Okay, anyhow, so option 2 cannot be answered, and as well option 4 also cannot be answered. Then chitinase. So chitinase can only degrade. So it cannot degrade only chitin. So chitinase is the enzyme, it can break down the chitin only. So means chitin is present in fungi. So fungal cell wall. So fungal cell wall can be digested by chitinase. Fungal cell wall can be digested by chitinase. Similarly, lipase break down the lipids. Cellulose is a polysaccharide, it is not an enzyme at all. So let us see the option 1. So lysozyme. So lysozyme is the important point we have to remember. Lysozyme. So lysozyme is the enzyme which degrades cell wall of bacteria. So lysozyme is the enzyme which degrades cell wall of the bacteria. So how it is degrading the cell wall of the bacteria? So this lysozyme break. So what it is doing? It is digesting. So it is digesting or breaking down. So digesting or breaking down. Breaking down of which part of the bacteria? Peptidoglycan. So peptidoglycan layer of bacteria. It is digesting peptidoglycan layer. So it is digesting the peptidoglycan layer of the bacteria. So we know that peptidoglycan is made up of N-acetyl muramic acid, N-acetyl glucosamine. So bond present between N-acetyl muramic acid and N-acetyl glucosamine will be broken down. So bond present between N-acetyl glucosamine and N-acetyl muramic acid will be broken down by this particular enzyme, that is the lysozyme. So main function of lysozyme is lysozyme breaks glycosidic bond. That is important point you have to remember. In multiple choice question they'll ask you generally. So what type of bond is broken down by the lysozyme? So lysozyme breaks the glycosidic bond. So peptidoglycan is a sugar. Peptidoglycan is made up of N-acetyl muramic acid is a sugar, N-acetyl glucosamine it is sugar. They both are joined in a peptidoglycan layer and they're broken. The bond present between this NAM and NAG is glycosidic bond, and this bond is broken down by the lysozyme. So anyhow, what is important point you have to remember is lysozyme degrade the cell wall of bacteria. How it is degrading cell wall of bacteria? It digests the peptidoglycan layer of the bacteria. And what type of bond it is breaking in the bacteria? Means it is breaking the glycosidic bond. So these are the few points which you have to remember actually. So lysozyme. So Ashwant is asking one question. So lysozyme is obtained from. So lysozyme is not obtained from bacteria. Lysozyme is actually obtained from. It is obtained from actually there is virus that is called as a bacteriophage. A virus particle that is called as a bacteriophage. Okay, so from bacteriophage you can obtain. So bacteriophage contains the lysozyme. So lysozyme can be extracted from the bacteriophage. Okay, remember that, because what is a bacteriophage? It is a virus particle which infects bacteria. So that virus particle, if it wants to infect bacteria, means it has to digest the cell wall of the bacteria. How it will digest the cell wall of bacteria? By releasing the enzyme lysozyme. So lysozyme is present in the bacteriophage. Very, very important point you have to remember. It is not part of the bacteria. Okay, it acts on bacteria. It is synthesized in the bacteriophage. Okay, and in human beings, so adhe man human beings తీసుకుంటే our saliva, మన saliva ఉంది కదా, saliva also contains lysozyme. So saliva also contains some amount of lysozyme. So that lysozyme which is present in saliva also digests the bacteria sometimes. So it is also playing an important role. Okay, so remember that saliva of the human beings also contain a small amount of lysozyme enzyme. Apart from that, bacteriophage is having the lysozyme. So these are the two sources of the lysozyme which you have to remember actually. Okay, so let us move to the next question. So Ashwant, you are asking why lysozyme is matched to bacteria means. So that's what I am telling you. Lysozyme degrades. Lysozyme is a digestive enzyme which break the cell wall of the bacteria. So that is the reason both are matching. Okay, lysozyme is the enzyme that is degrading the cell wall of bacteria. So it is both are matching. So that is the reason. So that is the match. So lysozyme is matched with bacteria. Okay. So let us move to the next question. Which of the following is a dual purpose codon? So if you see the first codon, so this is UUU. This is UUU is coding for actually the amino acid that is called as a phenylalanine. So it is coding for phenylalanine. So it is coding for an amino acid. The name of the amino acid is called as a phenylalanine. So AUG. So AUG is coding for, it is coding for methionine. So AUG is coding for methionine. At the same time, it is also acting as a start codon. So it is acting as a start codon. So as AUG is coding is a start codon, and it codes for amino acid that is methionine. So it is called as a dual purpose codon. So because of that reason, it is called as a dual purpose codon. So our answer will be AUG. So if you see UAA or UAG, so UGA, so these both are stop codons. So these are stop codons which stop the translation process. They don't code for any amino acids. So they don't code for any amino acids actually. So UAA, UAG, UGA. So both these three are called as stop codons. They don't code for amino acid. They stop the translation process. So in our question is asking which of the following is a dual purpose. So which of the following has two functions? Then so no doubt it will be AUG because it is a coding for an amino acid that is methionine. It also acts as a start codon. So that is the reason it is a dual purpose codon. Then UUU. So UUU code for the an amino acid that is called as a phenylalanine. So that is the answer to the students. So finally our answer is AUG will be our answer. So AUG will be our answer, students. Okay, let's move to the next question. Yes, welcome to Komla. So welcome to Unacademy EAMCET YouTube channel. So this week, this month I will be discussing the whole EAMCET question paper, students. So if you are preparing for next year medical entrance examination, try to join everyday classes. Okay, so last year if you are succeeded or okay, if you are not succeeded, go for the drop coaching or long term coaching or repeater batch, whatever maybe. So join the classes every day at 4 p.m. we'll be having biology class. So next month I will be taking the topics. So you can join the special classes also. So there is a description box is there. There is a link for the special classes. You can join those special classes also, free special classes. Still, if you are more interested, you can join plus courses also. Okay. So in plus course, I am taking the human physiology. So try to join, students. Free classes, you come and join. So if you are interested, later you can take the plus classes. Okay. So inka photoperiodism. So next question చూద్దాం. So in photoperiodism. So next question. So photoperiodism, the site for perception of light. or dark duration. So photoperiodism, term itself is telling photo. Okay, so photo means no doubt it is something related to the light. Okay, so in a plant, we know that in a plant we have stem, we have root, we have stem, so we have leaves, we have flowers and fruits. Okay, then what is important thing? Photoperiodism. So light. So light is influencing on the activity of the plant actually. So but light is received by which part of the plant? So photosynthesis. Photosynthesis is taking place especially in the more a maximum amount of photosynthesis is taking place in the leaves, and some amount is also taking place in the stem, because stem also have the some amount of chloroplast. So that is the reason in a plant parts, if you see photoperiodism, photoperiodism is exhibited by the leaves actually. So why photoperiodism is exhibited by leaves? Because the sunlight is trapped by the chlorophyll pigment that is present in the chloroplast. And where is the chloroplast is present? The chloroplast is present inside the mesophyll cells. And where is that mesophyll cells are present? Mesophyll cells are present in the internal in the leaves. So certainly the photoperiodism, okay, it is totally related to the leaves actually. So where the location where the trapping of sunlight is taking place. So in a plant, the location where maximum trapping of sunlight is taking place means it is inside the leaves. Inside the leaves where the trapping is taking place, inside the leaves we have mesophyll cells. Okay, inside mesophyll cells, within mesophyll cells we have cell organelle that is called as a chloroplast. Inside chloroplast we have chlorophyll pigment. Okay, that chlorophyll pigment will undergo reaction. That reaction is called as a light reaction. So that is the reason. So we have to take the answer as option 2 as answer. Okay, so like that we have to take the option 2 as answer. Okay, so that is about this question, students. Let us move to the next question. So let us see the next question. So match the following. Next question is, so term nucleon. So term nucleon was first time. So term nucleon was first time given by Frederick Mister. So Frederick Mister is a person so who has given the term nucleon. So term nucleon was given by Frederick Mister. Then semi-conservative DNA replication. So semi-conservative DNA replication was given by Watson and Crick. So Watson and Crick are the person who has got the Nobel Prize for explaining the 3D model of the DNA. So wait, there is a mistake. So double helical model is Watson and Crick. So semi-conservative replication is given by Measles and Stahl. So Watson and Crick has got the Nobel Prize for explaining the 3D model of the DNA. So that is 3D model is explaining that DNA is a double helical model. So DNA double helical model is given by Watson and Crick. Semi-conservative replication was given by Measles and Stahl's experiment. Then Lac-Operon. So Lac-Operon is given by Jacob and Monod. So Jacob and Monod is given the Lac-Operon. So based on that, let us see. So nucleon. So nucleon is given by nucleon was given by Frederick Mister. So A will be 3. So yes, here we can have 3 is there. Here also 3 is there. Then semi-conservative replication was given by Measles and Stahl. So here also 1 is there. Then double helical model of DNA was given by Watson and Crick. So C will be 2 and Lac-Operon is 4. So this will be our option, students. Okay. Yes. So very good. So formula. So option 3 will be our answer. So welcome to few more students. Welcome to Harita. Next question. So which of the following plant hormone acts as antagonistic? So antagonistic to gibberellic acid. So which of the plant hormone is acting antagonistic? So antagonistic means opposite. So which is acting opposite to the gibberellic acid? So opposite to gibberellic acid is abscisic acid. So abscisic acid promotes the closure of stomata. So what is that? It promotes the closure of stomata. Okay, so it helps in the closure of the stomata actually. So whereas gibberellic acid enhances the opening of stomata. So that option 3 will be the answer actually. Okay. Next question, students. So Melvin Kelvin has worked on which of the following? That is the question. So Melvin Kelvin is a scientist, no doubt about it. He is a scientist. he used the isotopes. He used isotopes. Okay, so he used isotopes in his studies. Okay, he used isotope in his studies actually. So, and his studies were conducted on carbon fixation. So in an organism that is called as a chlorella actually, he has conducted studies on the green algae, that is chlorella, and main purpose is to identify how the photosynthesis is taking place. So no doubt photosynthesis is also called as a carbon fixation. So carbon fixation means trapping of carbon dioxide is called as a carbon fixation. You have to remember this. Trapping of carbon dioxide is called as carbon fixation. Similarly, nitrogen fixation means atmospheric gaseous nitrogen is absorbed. That is called nitrogen fixation. So when carbon is taken, it is called carbon fixation. You have to remember this. And another important point what you have to remember is these fixation process are always a reduction process. Okay, remember that. So anyhow, so based on that we have to take the answer. Option 2 will be our answer. Next. So which of the following does not help in early diagnosis? So question is telling that early diagnosis. So if you see the PCR, so no doubt PCR is used in diagnosis, no doubt about it. So nowadays we are using PCR as a method for diagnosis, yes or no? Especially for the coronavirus. For coronavirus, so person who has infected with corona, so to confirm whether it is corona or not, we are doing some tests. So normal test, apart from normal test, the confirmation test is PCR test. By PCR only we are confirming. So no doubt PCR is a diagnostic test. It is not an early diagnostic. It is a diagnostic test. Similarly, ELISA is used to test the HIV virus. So no doubt it is also a diagnosis. It is also a diagnosis. It is also involved in diagnosis. It tests HIV virus. Okay. So then rDNA technology. So rDNA technology involves PCR and ELISA. Both are part of rDNA technology only. PCR and ELISA both are the part of rDNA technology. So that means rDNA technology is also used in the diagnosis. Then early diagnosis. So early diagnosis, serum analysis. So what is the serum analysis means? In the serum, a diseased person's serum is examined. Then what happens is, So in coronavirus is nowadays tested by using PCR, no doubt, and specifically it is RT-PCR. Very good. H1, it is RT-PCR, no doubt about it. RT-PCR means it is reverse transcriptase PCR actually. Reverse transcriptase polymerase chain reaction. Okay, RT-PCR it is called. So serum analysis. In serum analysis, what you will identify? You will only identify antibodies. Antibodies will be identified, but it is not the diagnostic process. Okay. So that is the reason. So we have to take the option A will be our answer. Okay. So and which are diagnosis, which are involved in diagnosis? PCR and as well ELISA. These are the diagnosis. Means they will confirm whether person is affected with corona or not. In ELISA they will confirm whether person is affected with HIV virus or not. That is diagnosis. Okay. So based on that we will take option A as our answer. Next. Okay. So we'll stop here, students. So our class is only for 30 minutes. Daily we will be having 30 minutes class only. So this week every day I will be discussing 10 to 15 multiple choice questions. Try to attend. So we'll discuss some points related to this multiple choice questions from next week. Okay. So means next month from November 1st we'll be taking some topics. I'll be discussing some topics. And you can join my classes on these special classes also. There is a description box is available. So in the description box, so you can identify the links or link for the special classes. You can join the special classes, students. They are also free classes only that are going on our app actually. So if you are preparing for next year medical entrance examination, I suggest you to take the plus classes, students. So there is 6 months plan is there. It is 10,125 and 12 months plan is also available. It is costing 12,375 rupees. So if you use my code RAJENDRALIVE, so that you can get the course for 10,125 rupees and 12 months course for 12,375 rupees, students. So thank you, students. Thank you all of you. So see you in the next class. So bye bye all of you.